So I want to start by reminding everyone when fatty acid synthesis is going to be occurring, and that's going to be again in the fed state when insulin levels are high. So in the fed state, when insulin levels are high, glucose is being taken up by the liver in an insulin-independent manner. And we'll see how this connects to fatty acid synthesis in the next couple slides. So the synthesis of fatty acids is an anabolic process that occurs in the cytosol of the liver under fed conditions. Remember, glucose is taken up under these conditions in the liver in an insulin-independent manner through GLUT2. And we have the processes of glycolysis, glycogen synthesis, the pentose phosphate pathway, the TCA cycle, cholesterol synthesis, and fatty acid synthesis occurring. And that's what we'll talk about today, fatty acid synthesis. So as glucose is taken up by the liver and flux through our TCA cycle, represented here only in part, will increase, what happens is that excess citrate can be shuttled out of the mitochondria to be used here as a substrate for fatty acid synthesis. To review quickly, we have pyruvate being produced through cytosolic glycolysis that enters the mitochondria and is converted to acetyl-CoA by our enzyme pyruvate dehydrogenase. Citrate synthase then converts acetyl-CoA to citrate, and here this is our substrate being shuttled out of the mitochondria into the cytosol, where it's initially cleaved by citrate lyase back into oxaloacetate and acetyl-CoA. The acetyl-CoA will be our substrate for fatty acid synthesis, and the oxaloacetate will then undergo a series of two reactions and will re-enter the mitochondria as pyruvate. So initially the oxaloacetate is reduced back to malate, and malate is decarboxylated to produce pyruvate. This reaction by malic enzyme is important as it generates NADPH. And in addition to requiring acetyl-CoA for fatty acid synthesis, we also need NADPH. So if we add here a few additional pathways, we can get a clearer picture of what's happening in this fed state metabolism. Again, we have glucose coming into the cell, which undergoes a series of reactions by glycolysis to generate pyruvate, which can enter into the mitochondria and is converted into acetyl-CoA. Acetyl-CoA is converted through the TCA cycle to citrate, and this is shuttled out of the mitochondria and is initially cleaved, as mentioned previously, by citrate lyase into oxaloacetate and acetyl-CoA. The oxaloacetate will undergo a series of reactions, first by malate dehydrogenase, as well as by malic enzyme, to generate NADPH, which is needed for fatty acid synthesis, which is shown here, our end product of fatty acid synthesis being palmitate. And these two sources, the pentose phosphate pathway and malic enzyme, are our primary sources of NADPH for fatty acid synthesis. So the first step of fatty acid synthesis after we have acetyl-CoA in the cytosol is the production of malonyl-CoA by our enzyme acetyl-CoA carboxylase. So the process of fatty acid synthesis starts with the carboxylation of acetyl-CoA to form malonyl-CoA. The enzyme involved, acetyl-CoA carboxylase, is also our regulatory enzyme. In addition to being the regulatory enzyme, it also requires biotin as a cofactor for this two-step reaction. Initially, in step 1, biotin is carboxylated, and in step 2, the acetyl-CoA is added to this bound carboxyl group and ultimately released to form malonyl-CoA. If we remember from beta oxidation, this malonyl-CoA in the cytosol is an important inhibitor of carnitine palmitoyl transferase 1. And this is important such that we don't have fatty acid synthesis occurring at the same time that we have beta oxidation. So illustrated here is our enzyme fatty acid synthase, and it's found as a homodimer. Each dimer consists of a single protein sequence that has 7 different domains of activity, which are shown here. Each of these domains will provide a specific reaction within the fatty acid synthesis. Most important are the acyl carrier protein, which is where we initially prime the fatty acid synthase with acetyl-CoA to initiate fatty acid synthesis. The subsequent series of reactions are carried out by these remaining 5 enzymes, which assists in both the addition of malonyl-CoA and subsequent dehydration reactions. And finally, after going through several iterations of synthesis where malonyl-CoA is added as a carbon unit, the product palmitate is freed from the enzyme through the activity of thioesterase. So if we look at the reactions catalyzed by fatty acid synthase complex, these are summarized here, and I'll draw your attention to some key parts. So initially, the synthesis of fatty acids by fatty acid synthase starts with the transfer of an acyl moiety, or acetyl-CoA, here, to the acyl carrier protein, or ACP, as noted in the previous figure. After this initial priming by acetyl-CoA, all additional carbon units are added in the form of malonyl-CoA to the elongating chain. So what you can see here is that this enzyme complex then transfers this malonyl-CoA to our acyl group in a subsequent decarboxylation reaction. And then we undergo a series of reactions of dehydration and reduction reactions, which require our NADPH. And the final product of this process, shown here, is palmitate. And this is after cycling through several iterations of adding malonyl-CoA to elongate our chain to a C16 carbon molecule. So what's also important to note about palmitate that we'll talk about in a couple weeks is that fatty acids are not stored in the liver. These are packaged into VLDL particles and transported to peripheral tissues for storage. And that's primarily the adipose. So to produce VLDL particles, these newly synthesized fatty acids are packaged into triacylglycerols. And tag synthesis can take place in both the liver and the adipose. So finally, how essential is fatty acid synthesis as a metabolic process? Well, after a quick literature search, it was found that fatty acid synthesis is essential in embryonic development. And if you remove fatty acid synthase, what happens is that any animals that lack the fatty acid synthase completely, they die in utero. And this is true in both mouse and humans. So I bring this to your attention to show you the online search engine OMIM, which is Online Mendelian Inheritance in Man. This is an online catalog of human genes and genetic disorders. The link for this reference is shown here. So if at any point in time in the course you're feeling curious as to what the relevance of an enzyme is and how clinically important it is, just pop it into this search engine. You'll get a review of literature as well as genetic locus of the gene to let you know what the signs and symptoms are if there's a deficiency of the particular enzyme you're looking at and how essential it is.